Assalamualaikum warahmatullahi wabarakatuh. My name Nurul Farha Zainuddin. I'm from University Teknologi Mara, Cawangan Perlis. Okay, this is my topic. I'll present about brain activity as a biomarker of cognitive processing and physiological responses. So basically, this is the content. I will talk about individual differences and cognitive processing, the biomarkers of cognitive, and the biomarkers of neurophysiology. So this is actually our short experiment. We have a preliminary findings of individual differences. So basically, this research or these experiments we conduct towards the five subjects, and we measure their IQ, and this IQ actually measuring the abstract reasoning. So they are actually almost similar age, and we conduct in the frontal area FP1 and FP2, so we have their ranking score. So based on the result, we find out that in the alpha power they have a significant difference of the variances between subjects. In here we can find out that subject C, which is at the last rank, he actually have a difference, a significant difference compared to the subject A, subject B, subject D, and subject E, which is we conclude that there is a significant difference between subjects for the alpha power. Meanwhile, for the beta power, we find out that subject C again, he have a significant difference between the subject A, subject B, and the subject D. So it also showed that there is a significant difference between subjects. So it showed that there is a individual differences, not only in terms of the cognitive ability, but also in terms of the neural activity. So the cognitive ability will reflects the rhythm of the neural. Alright, so we can interpret from the findings based on the results. The main findings of this preliminary study is to examine the variances between subjects in different level of difficulty, reveal positive findings for further in-depth justification or exploration toward intelligence between high scoring and lower scoring subjects. Because of there is a significant difference of the subject C, which is at the lowest scoring subjects. It also provides the reference for the investigation at frontal cortex, especially in the area of FP1 and FP2, as a function for the intelligence element of reasoning, because of at the frontal area, the responsibility for the frontal area of our brain is actually for the cognitive processing, okay, the decision making, those things. Okay, in regards with the individual differences, it also actually reflects to the neural efficiency. Okay, because of individual differences, they also have a difference in terms of the neural efficiency. It also known as a neural efficiency hypothesis. Alright, so we can see based on this result, ERD result, we can find out that they have a difference of the high IQ brain and the low IQ brain. So this actually defined as the high intelligence individuals. They show had a lower brain activation than less intelligence individual when working on the same cognitive task. So if they are working on the same cognitive task, the high IQ and the low IQ have the different brain activation. So in high IQ, they have a less activation compared to low IQ. So what does it show is actually okay, that one is based on the color. So it showed that brain of individual with higher intelligence, they actually utilize less glucose. So those with higher intelligence that portray lower brain activation, they actually utilize less glucose during the abstract reasoning and attention task, during this kind of task. This is the extended biomarkers of the neural efficiency theory. So because of the activation, okay, the interaction of the neural activation and the cognitive ability actually is moderated by the task difficulty. Okay, apart from task difficulty, they also have another factors such as gender, task type, task difficulty, and brain area. So based on this figure, it showed that more intelligent individuals show lower brain activity than less intelligent only when working on cognitive tasks at low to moderate level of difficulty. So based on this graph in here, NE neural efficiency, the high IQ actually show low neural activation compared to the low IQ. Alright. Only when working on the cognitive task at low to moderate, okay, only when working on the task at low to moderate. Alright, so this is based on the level of the task difficulty. They can actually moderate this different kind of result. So in measuring neural activity, we have a few instrumentation. The most common instruments measuring neural activity in cognitive is actually fMRI, EEG, and functional nears. Okay, so we are using EEG. Okay, but if you wanted to know the blood glucose mechanisms, the in terms of the biochemistry mechanism, we can use fMRI or the functional nears. Okay, this is a quick info about the neuroplasticity. Okay, so that figure actually showed the capacity of the neuron and the neural network is in the brain to change their connection. Okay, so this is what happened to the brain when they find when they have a new information or the new cognitive processing. The brain actually tend to rewire the connection. Okay, because of they actually growing in terms of the neural capacity because of that neural information. Okay, so this is what happen when a person train their brain, when a person uh, keep working on something, keep improving their cognitive ability, the connection of the neural activity, like rewiring the connection to have a more great capacity of the cognitive processing or the brain processing. So this is actually another biomarkers of brain activity. They converted to the frequency band. We have 5 different frequency band, which is a delta, theta, alpha, beta, and gamma. So delta theta is actually using a low frequency. So usually we try to measure the neural activity in a condition of the sleep condition or the awakened condition. For the alpha frequency, we usually measure in terms of the arousal. Okay, based on previous findings, based on previous research, they measure arousal. They wanted to measure um, a relaxation. Okay, contradict to the arousal and attention. For the beta activities, based on previous findings, they wanted to know a condition in more higher of cognitive processing or with the sensory motor processing presents, and then as well as a gamma it also wanted to know uh, mm, very high level of the higher order of cognitive processing. All right. And then this is biomarkers of alpha, beta wave, and neural efficiency. Neural efficiency in here actually represented by the alpha beta ratio. Okay, so this is another physiological biomarkers for the cognitive processing. For the alpha, they measure in the condition of the fatigue, relaxation, and the arousal. Based on previous findings, for the beta, they used to measure the condition of the involvement of the sensory motor processing. So if we move on something, if we moving like we are conducting um, a psychomotor or any motor task, we will use the beta frequency to measure the neural activity. Okay, so for the neural efficiency that represented by the alpha beta ratio, we usually wanted to know the vigilance, the alertness, and sometimes we will also wanted to know the high order kind of processing during the very difficult task, okay, or the very demanding task. Okay, as for the conclusions, the individual differences intelligence leads to the individual capability to generally cope with challenging situation. So, in order to understand human cognition in a subjective manner, psychometric tasks are the most commonly used tool. But if we wanted to more objective manner, in an objective manner, we will use the instrument of the current technology, which is we call it as a EEG functional awareness that I presented before. In fact, neural activity was also employed to understand the brain mechanism physiologically. If we use the current technology, we can measure the neural activities, the brain mechanisms based on the location, okay, based on the interaction of the cognitive. So that's all my presentation. Thank you. So I'll open for the questions.